2020, and this is week 13. We earlier this morning we did the first 14 slides of the medical microbiology intro, how to be a good pathogen, where we describe some virulence factors, and we left off at slide 14 talking about facultative intracellular parasites. And so what we're going to do for part 2 of virulence factors, we're going to start at slide 15. So let's see if we can get this set up here. Okay, oops, that's not what we wanted. Okay, so folks, so for part 2 of virulence factors, we're going to use kind of the king of the invasive bacterial pathogen, Staphylococcus aureus. So folks, just really quickly remember Staphylococcus aureus, gram-positive coccus. It causes beta hemolysis on blood agar. It can grow on mannitol salt agar and ferment mannitol, turning the agar yellow. It's going to be catalase positive. So those are some of the traits that you might see on our lab exam next week. Okay. So why is Staphylococcus aureus such a brilliant tissue invader? Like you could start out with just maybe a little infected site on your skin, and within a couple of days, potentially the staph has spread everywhere. And I know that we were always told that Staph aureus is brilliant at making little tiny micro abscesses, little abscesses throughout your body. So just by looking at just a few of the Staphylococcus aureus virulence factors, and again, folks, there's tables of Staph aureus virulence factors, we're going to see if we can understand how Staphylococcus aureus, it can coordinate its attack based on its population size. So we'll introduce this really cool phenomenon called quorum sensing, where the bacteria can sense their population size and then turn on and turn off different genes depending on their population size. And we'll see how Staphylococcus aureus can use quorum sensing to help avoid our immune response and therefore spread throughout the body. So I'll go through this one step at a time, folks. And again, if you could look at your virulence factor worksheet that we posted on Canvas, you'll see that most of these virulence factors are on that worksheet. So certainly you'd want to be able to describe what they do, how they act for lecture exam 3. Okay, so you guys will first start with hyaluronidase, right here, hyaluronidase, and this is a little cartoon here. So let's say that these are epithelial cells of a human, right? And let's say this is our Staphylococcus aureus. Well, to get into deeper tissues, the bacteria have to move between these epithelial cells. And animals have evolved ways to prevent bacteria from moving between their epithelial cells. So we have little protein rivets that hold these epithelial cells together. And furthermore, the cells will produce a substance, we'll call it intercellular cement. Okay, so we have rivets and cement trying to hold these little cells together. Well, what does Staph aureus do? It makes an enzyme called hyaluronidase. And what the hyaluronidase does, it digests the intercellular cement. And that means then Staph aureus can move between the epithelial cells into deeper and deeper tissues. Hyaluronidase, kind of a common name for it, is spreading factor, right? So this is one reason why we start with one little infected site in our bodies, and then in 24 or 48 hours, Staph aureus could spread everywhere because of this powerful hyaluronidase. Okay, and then folks, what we're going to do is we're going to go over here, and this is when we're going to get into quorum sensing. So let's say Staph aureus has just invaded us, and so let's just say, you guys, these little purple balls here is a Staph aureus. When the Staph aureus population size is small, there's not very many of them, they really need to find a way to protect themselves from host phagocytic cells. So the Staph aureus will make this neat enzyme called coagulase, and it sounds like coagulation, right? Blood clotting. And what coagulase does is it takes fibrinogen, which is a substance we make, and converts it into fibrin. And these little lines right here, you guys, I think of these as these little strands of fibrin, and fibrin is part of a blood clot. And what the fibrin is going to do is it's going to lay down a protective wall around the Staph aureus. So I think of this as the little fibrin fort that's going to protect the Staph aureus when they're in low numbers from phagocytic cells, for example, right? So this might be a little micro abscess. So the Staph aureus are really happy, they're protected inside. They're growing and dividing, growing and dividing, growing and dividing. So you can imagine that pretty soon, with the population explosion of Staph aureus within that little fibrin fort, now it's no longer protective, it's actually become a fibrin prison, right? And conditions within the little abscess are horrible, right? Lots of toxic metabolic waste products, maybe not a lot of food. So now that the Staph aureus, the population size is really large, what the Staph aureus wants to do is basically a prison break. It wants to break out of what's become its little fibrin prison. But to do that, it has to turn off expression of the coagulase gene, right? It has to stop making coagulase. It doesn't want more fibrin to be laid down. So it has to turn off expression of the coagulase gene, and then it needs to turn on expression of a new gene called staphylokinase. And the role of staphylokinase is to break down the fibrin, right? Break down the prison walls, and that will permit the Staph aureus to escape. Now, this is pretty amazing, right? So how can the Staph aureus know when to turn off coagulase and when to turn on kinase? Well, again, folks, this is this amazing phenomenon called quorum sensing. So what the bacteria do is they produce these little, each of the Staph aureus will produce a chemical, and we'll just call it an autoinducer. And then the Staph aureus have receptors, so they can sense, quote unquote, the concentration of autoinducer. So when there's not very many Staph aureus present, the autoinducers will be in low concentrations, and that permits expression, transcription, and translation of the coagulase gene. So when Staph aureus is in low numbers, there's low autoinducers, so the coagulase gene will be expressed. When the Staph aureus grows to really high population size, we have a massive amounts of autoinducer. And when enough autoinducer binds to the receptors on the Staph aureus through a complicated series of events, it will result in transcription of the coagulase gene being shut off. And then we're going to turn on transcription of the staphylokinase gene. And again, this is based on population size, and it's just brilliant. So we want to remember then, folks, that it's through quorum sensing that Staph aureus can sense its population size. It can turn on the coagulase transcription when the population size is low. It will have turned off, would not be expressing staphylokinase. And then when the number of cells increases, they'll, through the autoinducers and quorum sensing, they'll turn off coagulase production and turn on staphylokinase production. And that's what lets them break out of their fibrin fort or prison only when their population size is really high. Now, why is that helpful? There could be lots of very frustrated phagocytes waiting around outside, ready to gobble up the Staph aureus when they break out. But since the Staph aureus has waited until the population size is really high, right? When they break out, some of them might be killed, but because there's so many, for certainly some of them are going to escape, maybe through the blood vessels or through surrounding tissues. And again, they're going to spread and start this process all over again, forming the little fibrin forts, the micro abscesses, and just repeat this over and over. So we can see how Staph aureus can spread really, really quickly through the body. So again, folks, it's the autoinducers and quorum sensing that controls the expression, the transcription of the virulence gene for coagulase and staphylokinase. Pretty amazing. And we also, folks, we want to remember that staphylokinase, excuse me, I said that wrong, you guys. Staphylococcus aureus is a pyogenic bacterium, so we would also predict that a lot of leukocytins would be released. Hopefully, well, for the Staph aureus, hopefully sufficient enough to kill a lot of the white blood cells in that area, so we get this nice accumulation of pus in our abscess. Okay. So we've been talking about how the bacteria initially avoid or evade our immune responses, right? So they can get colonized. So now we want to take a look at why we call these microbial diseases diseases, right? So dis-ease, your body is not at ease. So we think of dis-ease as an upset in the normal homeostasis of our body. So things aren't functioning properly. And there's different ways that microbes can cause damage. We're going to see through inflammation and in general inappropriate immune reactions. Remember the double-edged sword of the immune system. We can't live without it, but often, especially we're talking about in viral infections, often it's the immune response that's going to cause a lot of damage to the host. And then, folks, we'll spend quite a bit of time talking about different types of toxins that bacteria can produce. Toxins, another name would be poison. And so we'll talk about the protein exotoxins, and then the gram-negative endotoxin, the lipid A of lipopolysaccharide. And generally speaking, the reason that the host will experience disease is we're going to have an inhibition of normal cell function, right? And if our cells can't function properly, that's going to be really serious for us and could potentially even kill us. Okay, so just so you know, these photos, you guys, this is anthrax, cutaneous anthrax. So probably this person had a wound in the shoulder here, and then endospores from the dirt, anthrax endospores got in here, they germinated, and from all the toxins and inflammation, we get these eschars, I believe they're called. And this is, oh my gosh, what a case of varicella or chickenpox this is, so caused by a virus. And this is infant botulism, folks, which we will be talking about. All right, folks. So again, if you look on Canvas, the virulence worksheet is one page, and the second page is a table where you are going to compare and contrast bacterial exotoxins to bacterial endotoxins. So that compare and contrast type table, folks, if we were doing a face-to-face exam, it would be a short answer question. You'd be filling in that table. But because we're going to be doing this online, just be aware I can still ask you a lot of questions with compare and contrast bacterial exotoxins to bacterial endotoxins, right? So the exotoxins, just really briefly, guys, they're made of proteins, and they have, there's a great variety of exotoxins, and they usually have specific cell targets, so we can't talk about a general sign or symptom for bacterial exotoxins. Now, in contrast, bacterial endotoxins, we know that the endotoxin is the lipid A of the lipopolysaccharide of the outer membrane of gram-negative bacteria cell walls. So we can talk generally about the signs, the symptoms, the damage that endotoxins are going to trigger. And we'll see, folks, that the endotoxins, they cause all their damage through triggering our cells to release too many chemical messengers. And the chemical messengers, you guys, that we'll just say generically help to coordinate cells of the immune system, we're going to call them cytokines. And the thing is, we want small amounts, low amounts of cytokines released locally, right, where the microbe invades us. But we don't want massive amounts of cytokines released that flood throughout our body. When we have too many cytokines released, they sometimes call it a cytokine storm, that can kill us, right? And it's the major event in what's called sepsis, or septic shock. And indeed, you guys, even though we're focused on bacteria, it's believed that maybe the reason the COVID, the pandemic coronavirus, is so deadly in some people is that the coronavirus is triggering this massive cytokine release. So people are dying of sepsis or septic shock. Okay. So folks, before we do compare and contrast, we have to learn some vocabulary. And the vocabulary we're going to learn is toxoids and antitoxins. This is related to exotoxins only. We're going to see endotoxins, you can't make toxoids, and with endotoxins, you can't make antitoxins. So let's just take this one step at a time. So what are toxoids? So toxoids are modified toxins. So we take the toxin and we're going to modify it, we're going to change it. So it can no longer cause harm, that's really important, yet it can still trigger a protective immune response. For example, the toxoid can still trigger production of protective antibodies. For example, neutralizing antibodies, it could bind to the toxin and block it from attaching to our cells, right? So neutralizing antibodies is one example. And what's so powerful, you guys, about toxoids is that we can use them in vaccines to protect our children, protect ourselves against some of the horrible exotoxin diseases that we'll be learning about. So as an example, folks, this is a really common childhood vaccine, the DTaP. The D stands for diphtheria, the T stands for tetanus, and AP stands for acellular pertussis. So in this, I guess you'd call it trivalent vaccine. There's 3 different vaccines in here. The D is for diphtheria toxoid, protects us against diphtheria toxin. The T is tetanus toxoid, protects us against tetanus toxin. And the AP, acellular pertussis, this is going to be the pertussis toxoid. And again, it will protect us against the harmful effects of pertussis toxin. So this vaccine, folks, will protect us against diphtheria, tetanus, and pertussis, right? So really powerful, really helpful vaccine. Now, the antibodies, the protective antibodies that we want to make against the toxins, those antibodies have a fancy name. They're called antitoxin. They're antibodies against the toxins. And again, folks, like one example, specific example would be antitoxin would consist of some neutralizing antibodies. And remember, neutralizing antibodies, they bind to, in this case, the toxin and prevent the toxin from attaching to the surface of our cells. And with exotoxins, if the exotoxin can't bind to our cells, the toxin can't get into our cell and therefore it can cause no harm. So here, folks, this would be, this is similar, probably not exactly like the endotoxin virulence factor sheet on Canvas, but the basic information is here. So you guys, so remember, we're going to compare and contrast bacterial exotoxins to endotoxins. So first of all, let's ask ourselves which bacteria make these toxins. So with exotoxins, these are proteins. So a bacterium simply has to have the gene for the protein, and then it can make the exotoxin. So we would say that both gram positives and gram negatives can make exotoxins. In contrast, you guys remember endotoxins. Endotoxins are the lipid A of lipopolysaccharide found in the outer membrane of the gram-negative bacterial cell wall. So only gram-negative bacteria can make endotoxins, right? Okay. Composition means what's the toxin made of. So remember, you guys, exotoxins are made of proteins. Endotoxins are the lipid A of lipopolysaccharide. Where are the toxins located? So usually, not always, but usually when the bacteria are alive, they're usually making and then secreting the exotoxin into their environment. There's some exceptions, but in general, you guys will say that the live bacteria are secreting the exotoxin from their cells into the environment. In contrast, you guys, the endotoxins are actually a part of the gram-negative cell wall, right? Part of the outer membrane. So you don't get a lot of endotoxin released until the bacterium dies and it lyses, and that's when we get lots of endotoxin release. With regard to toxicity, we could think of like how dangerous is the toxin, and so one way to compare this is to say, well, does it take a tiny amount of exotoxin to cause great harm? And the answer is yes. So exotoxins tend to be highly toxic. It can take just tiny amounts, like nanogram amounts, maybe to kill, say, a rodent. But in contrast, you guys, the endotoxins in low concentration, low amounts of endotoxins, usually that isn't going to cause a lot of harm. It's when large amounts are released. So, for example, you have bacteria growing in your bloodstream, and the bacteria start to die and lyse and release the endotoxin. That's when we're going to get those massive cytokine releases. Okay, and again, the endotoxin can be rapidly fatal when large amounts are released, when the gram-negative bacteria die. Signs and symptoms, remember you guys, with exotoxins, there's such a variety of exotoxins, and the signs and symptoms are toxin specific, so there's variable effects. As a class, they are not pyrogenic. And pyrogenic, folks, if you think of a pyromaniac likes to set fires, right? So in pyrogenic, we're talking about fever production, the internal fire. Okay, so in general, exotoxins are not pyrogenic. There's some exceptions, but generally speaking, they're not pyrogenic. They won't trigger a fever. In contrast, you guys, remember the endotoxins are going to cause the same cytokine storms. So we're going to see the same clinical signs and symptoms regardless of which gram-negative bacteria are causing it. So endotoxins, you guys, they are pyrogens, so they're going to trigger a fever. And as a result of that massive cytokine release, that cytokine storm, we have changes in our blood vessels, which are really bad. The blood vessels dilate and they become leaky. So we start to lose the liquid portion of blood plasma from the blood vessel itself. That fluid moves into the extravascular tissues. So the combination of vasodilation and capillary permeability are leaky vessels. You get low blood pressure or hypotension. And when you're hypotensive, there's not sufficient blood pressure to make sure that you get sufficient blood delivery to your organs and your tissues, right? So this is cutting down blood flow to your tissues, to your organs. Another horrible thing as a result of those cytokines that are released is you trigger your clotting cascade. So now you're going to start throwing little blood clots everywhere to further block blood flow. And that phenomenon is known as DIC, and it stands for disseminated, means spread throughout the body. Intravascular means within the blood vessels. Coagulation, so clotting. So again, in DIC, we're throwing little blood clots everywhere throughout the body, further decreasing blood flow to our organs and tissues. So as a result, we're going to end up with multiple organ system failure. Your kidneys start shutting down, your liver starts shutting down, your brain starts shutting down, right? You might become confused and incoherent, right? Lungs can't work, right? And we know that that rapidly progresses to shock, and very often it can lead to death. And these stages can happen so quickly, you guys. It's really scary. And sometimes this collection of events, it's referred to as gram-negative sepsis or septic shock. Maybe you've heard the term septicemia. Septicemia in general means when you have a poison in your blood, and one cause might be gram-negative bacteria growing in your blood, causing the massive release of cytokines. And again, you guys, this can be fatal. With regard to heat sensitivity, since the exotoxins are proteins, they can be natured by heat, so we'd say they're heat sensitive or labile. The one exception, you guys, is that doggone Staphylococcus aureus can make a exotoxin called endotoxin that its target is the intestinal cells, and it is relatively heat stable. So you might recall that we said if Staph aureus was growing in a food and making the endotoxin, like salted meats, for example, Staph aureus is growing in the salty meats, making the endotoxin. If you don't heat the food sufficiently, or maybe don't heat it at all, right? And even if you did heat it, again, that endotoxin is relatively heat stable. When you swallow the staph endotoxin, you have that projectile vomiting, and it is dramatic and it happens really quickly. So in general, folks, again, exotoxins in general, they're heat sensitive or heat labile, meaning if we heat them, we'll denature them and they can no longer cause harm. Now, in contrast, you guys, this is bad news. That lipid A of the endotoxin, it is heat stable. It's resistant to heat. You can autoclave the endotoxins and they will not be inactivated. So that's really important. If you think you have contamination, say, of a bag of saline, right? You're like, oh, I'll just autoclave it. But if there's gram-negative bacteria in there, the gram-negative bacteria will die, but the endotoxin will still be active. So if you infuse that contaminated saline into a patient, you could throw them into endotoxic shock. So folks, remember the toxoid. This is the modified toxin. We can use it as a vaccine to trigger production of protective antibodies. And those protective antibodies against the toxin are called antitoxin. So what we're going to do now is ask ourselves, can we make toxoids against exotoxins? Can we make antitoxins against exotoxin? The answer is yes. So you can make toxoids from the exotoxins, and you can make protective antitoxins against the toxoids. Bad news, you guys, on endotoxins, you can't make toxoids from the lipid A. Lipids are just lousy antigens, so you can't make a toxoid out of them, and you can't get protective antitoxin made. So this is a real challenge, right? If you have a patient going into gram-negative sepsis or septic shock. So, folks, we'll just take a look at a couple of examples of bacterial exotoxins, and on Canvas, I posted a scan of some pages out of a medical microbiology book on tetanus toxin and botulinum toxin. I often like to ask questions about those two toxins on lecture exam 3, and usually on our comprehensive final exam, there'll be a clinical case that will deal with either a tetanus clinical case or a botulinum clinical case, or probably both. So those are two toxin-mediated bacterial diseases that you'd want to know for lecture exam 3 and for our final exam. Okay, so the two toxins we're going to describe, you guys, are botulinum toxin, which causes botulism, and tetanus toxin, which causes tetanus. And in this case, you guys, we have babies, like neonates. So this is neonatal tetanus, and this is called infant botulism. Okay, so both of these toxins or exotoxins are made of proteins. They're strongly antigenic, right? So we're going to have a strong immune response. And the good news is, you guys, you can make toxoids from them. Now, what I don't understand is why historically we have a tetanus toxoid vaccine, but we don't have a botulinum toxoid vaccine. I'm not sure why. Okay, but we have a vaccine to prevent tetanus. We don't have a vaccine to prevent botulism. Okay, so what's so fascinating is these two toxins are made by two cousins. So the bacterium that makes botulinum toxin is called Clostridium botulinum, gram positive, strict or obligate anaerobe, right? They can't grow in the presence of oxygen because they never evolved those protective enzymes. And then its cousin, Clostridium tetani, makes tetanus toxin. So two species of the same genus, Clostridium, and again, the Clostridium tetani makes endospores. But what's so fascinating, you guys, if you look at the amino acid sequence of the botulinum toxin and compare it to the amino acid sequence of the tetanus exotoxin, they're almost identical. They're really, really similar with just a few variations in amino acids. And furthermore, what makes it fascinating is that botulinum toxin, it acts in the peripheral nervous system. So those of you that have had ANP, I know this will make sense, and those of you that haven't, try not to stress too much about this. But botulinum acts, as we said, in the peripheral nervous system where motor neurons innervate skeletal muscles. And what the botulinum toxin does, it inhibits the release of what's called a neurotransmitter that's going to be made by that motor neuron. And so whenever the motor neuron is carrying what we call a nerve impulse or an action potential, at the point where the motor neuron innervates the muscle, the neuron should release acetylcholine. It will diffuse across and bind to the surface of the muscle cells, and that will trigger this amazing cascade of events that will lead to muscle contraction, right? So, when the botulinum toxin is absorbed into the motor neurons, they prevent the release of the acetylcholine. And that means our muscles can't contract, right? And so as a result, you end up with what's called a flaccid paralysis or a ragdoll paralysis. See this little baby, just like a ragdoll, poor little baby. And what kills us is that the muscles involved in breathing are paralyzed, so we suffocate. What a horrible way to go, right? Now, so historically, there's actually three ways that we could be affected by botulinum toxin. So historically, probably the most common way was when we used to do a lot more home canning, when more of us were being raised on farms. You know, we had our own gardens, our own vegetable gardens, and it was a lot more common for people to can their own vegetables, right? So you'd maybe harvest your vegetables in the summer and then you'd can them so that you'd have them available to eat in the wintertime. The problem with Clostridium botulinum is that if you do your harvest of your veggies and put them in, say, your mason jars, and there's botulinum endospores from the soil, right where the vegetables grew, and if you don't heat your jars or your cans to 121 degrees Celsius, 15 pounds per square inch of pressure for 15 to 20 minutes, those endospores won't be destroyed, right? They're really, really resistant. And do remember, you guys, one reason the autoclaves were developed is to destroy endospores. Normal autoclaving will destroy endospores. But if you're just a home canner, right, maybe just boiling things on your stove, you might not destroy the endospores. And this is a big problem, especially if you're canning low acid vegetables, like green beans are the classic ones, right? Green beans don't have a lot of acid. So Clostridium botulinum then, in those improperly canned green beans, the endospores will germinate because eventually the environment becomes anaerobic. They germinate, they grow, and they start making lots and lots of botulinum toxin. Now remember, the botulinum toxin, exotoxin, it's a protein. So if you pulled the green beans off your shelf and you heated it up really well, right, you could denature the botulinum exotoxin. But if you didn't heat the green beans, let's say you put them on a salad, so you didn't even cook them, right, and you started eating them, you'd be swallowing botulinum exotoxin, and you'd end up with a flaccid paralysis, right? So we know with adults, a lot of home canned, like we said, like green beans, kind of alkaline vegetables, we can end up with an intoxication, right, where we're actually swallowing the preformed toxin. It's what we just described is not an infection, it's an intoxication. But in contrast, you guys, in infant botulism, this is really important for us to know. They associate infant botulism with babies, and I think the cutoff age is 18 months of age. So babies 18 months of age or younger, if we give them honey, and if you think about this, you guys, so where does the honey come from? It comes from honeybees, right? And the honeybees go and they visit the flowers and gather nectar and pollen. And the flowers, right? The flowers, the plants, germinated from the soil. So it's possible that the flowers and the pollen might be contaminated with botulinum endospores. So the bees bring it back to the cave. The cave. Oh my goodness. The bees bring the nectar and pollen back to their hive. And so then the endospores are going to get concentrated in the honey. So then if we give the honey to our little babies, less than 18 months of age, they swallow the endospores, and the endospores can germinate in the anaerobic environment of the intestine. Now, when I was going to college, they said the reason that babies suffer from this ingestiform is because the pH of the baby's digestive system isn't low enough. So Clostridium botulinum aren't supposed to grow at low pH, right? And then now we know, you guys, well, it might have to do with the baby's good normal intestinal microbiome hasn't developed fully, right? So it might be the good guys in the intestine help to lower the pH in the intestine, and certainly those good bacterial mutualists in the intestine would compete with Clostridium botulinum, so they couldn't grow to large numbers. So in these poor babies, then botulism is actually an infection. The Clostridium botulinum is growing in their intestine, pumping out the botulinum toxin. So the important thing is that if the baby needs to get medical care, be intubated right to get them through the respiratory arrest. Eventually, the botulinum toxin will break down, right? You just need to be able to supply supportive measures for the baby. So put them on a ventilator, and you can pull a baby through as long as you get them to the hospital in time. A third type of botulism some of you might encounter is called wound botulism. In the old days before antibiotics, wound botulism would be a common consequence of going into battle, because you can imagine, people fighting with swords or sabers or cannonballs or um, with bullets, that those wounds would literally get dirt in them. They would get dirty, right? And what's in the dirt? Clostridium endospores. So when there's any kind of a wound, there's damage to the blood vessels. So you end up with no blood flow. So you can end up with a little anaerobic area there. So the Clostridium botulinum endospores can germinate, start pumping out botulinum toxin. So you end up with wound botulism. Well, with the discovery of surgical asepsis and antibiotics, usually that shouldn't be a problem in warfare anymore. But where you will see it nowadays is in your patients that are using IV drugs and don't have access to clean needles and syringes. So what happens is the needles and syringes, they might be used over and over again, and literally, you guys, they can get dirty. And so if, say, a person has been shooting up and they have a lot of abscesses on their arms, right? They have a lot of infections. If they shoot up into one of those abscess areas where there's no blood flow, right? And the needle has the botulinum endospores on it, the endospores will germinate in that anaerobic area, and they'll end up with wound botulism. So it's something to remember in the emergency room, right? That you might see wound botulism in IV drug users who don't have access to good, to clean needles and syringes. And then, folks, we're going to go down here to tetanus. And again, this little baby is suffering with neonatal newborn tetanus. And it's fascinating. So let's do the newborn tetanus first, you guys. So when the baby's born, remember the umbilical stump will be up here. The umbilical stump, after it's cut, there's no blood supply. So until that stump falls off, that's an anaerobic area, right? So if the instruments that are used to cut the umbilical cord literally are dirty, right? Think of pioneer days, when maybe Pa would pull out his hunting knife to cut the umbilical cord of his newborn baby. If the instrument is contaminated with dirt and has Clostridium tetani endospores in it, the umbilical stump then gets seeded with those Clostridium tetani endospores. And in that anaerobic umbilical stump, the endospores germinate, the Clostridium tetani start growing, start pumping out tetanus toxin. Now, tetanus toxin, you guys, is wild because it works at the central nervous system. And what it does is it stops the release of what we'll just call generically, you guys, inhibitory neurotransmitters. And as a result, and those of you in A&P, I know this is not a very good explanation here, but the result is all of your skeletal muscles contract at the same time, right? And this is called spastic paralysis. And you can see, you guys, this poor little baby, it's like a board, right? You would never hold a little baby just by its head, right? Because the neck muscles are so weak. But in tetanus, the muscles, every muscle is contracting as hard as it can. So we end up with almost like this, almost like a board here. And again, the reason we would die is from respiratory failure, right? The muscles involved in breathing are paralyzed, and what a horrible way to go. Now, the thing here, folks, is if this little baby's mom had been current on her tetanus vaccines, the good thing, if mom is vaccinated, mom can pass her protective neutralizing antibodies, and it's a specific class, you guys, that we'll talk about later called IgG. So mom's maternal antibody can cross the placenta and enter the baby's bloodstream. So when the baby is born, the babies would have circulating maternal antibodies from all the vaccines the mom has ever had and all the pathogens mom has ever encountered. So one way to protect babies from neonatal tetanus is to make sure the mom has been vaccinated against tetanus. She can provide passive immunity, provide her maternal antibodies to the baby, and those maternal neutralizing antibodies would bind to the tetanus exotoxin and stop it from binding to the baby's neurons, right? And then the baby wouldn't experience neonatal tetanus. Okay? Now, the other way we can get tetanus, you can get the classic one when we were growing up was to be running around outside without our shoes on in the summer and step on the proverbial rusty nail, right? And my mom would tell me, if you run around without your shoes and you'll step on a rusty nail and then you might die of lockjaw, right? So lockjaw is the common name for tetanus. And there was real wisdom in that, you guys, because think of a rusty nail. It's out in the dirt, right? So it's been contaminated with dirt. They could have the Clostridium tetani endospores. If you step on it, you get this deep puncture wound, right? So the endospores are injected deep into your tissue. The nail itself damages blood vessels. So now the endospores are in this nice anaerobic little pocket. So the tetanus endospores can germinate. Clostridium tetani grows, starts pumping out tetanus toxin, and then indeed, just like your mom told you, you could die of lockjaw, right? The good thing is, though, you guys, right? We have the tetanus vaccine, right? So we want to make sure that our kids are immunized, and you need a booster every 10 years. And very often, young people your age, college age, you've fallen behind on your tetanus boosters. So the next time you get an opportunity, you guys, double check and make sure that you're current on your tetanus booster, right? Because you don't want to develop tetanus, that's for sure. Okay, so tetanus causes spastic paralysis. Botulism causes flaccid paralysis, and both types of paralysis will lead to respiratory failure and death from suffocation. In both these cases, you guys, just one last thing. Like for both these babies, if they're presented at the hospital, there's antibodies against the botulism toxin. Remember the antitoxin? So you can give antibodies against the botulism toxin to help this little baby, right? And likewise, you guys, we have antibodies against the tetanus toxin. So we could provide passive immunity to this poor little baby, providing tetanus antitoxin. Still on exotoxins, you guys, many of the protein exotoxins are what we call AB toxins. And an AB toxin has two different subunits, an A subunit and a B subunit. So let's talk about the B subunit first, because B stands for binding. So this is the part of the toxin that binds to a specific host cell surface receptor, right? So if you could somehow block the B subunit from binding to the host cell, the A subunit, which is the mischief maker, the trouble, this is the active subunit. If B doesn't bind, A can't get into the cell, and it's the A that's going to cause the disruption in normal function within the host cell. So if you have a vaccine, you could just make it out of pure B subunit. You vaccinate your patients, they make antibodies against the B subunit, so if they ever encounter the real AB toxin, right, you'll have those neutralizing antibodies will bind to the B subunit, block its attachment from host cells, and then the A subunit can't get inside and can't cause damage, right? That's kind of cool. And then, folks, just really quickly, we're kind of repeating ourselves here, but again, just to again, contrast the bacterial endotoxins. So remember, you guys, the bacterial endotoxin, it's the lipid A of lipopolysaccharide of the outer membrane of gram-negative cell walls. Lipids are horrible antigens, you guys, meaning we don't get a strong protective immune response. And furthermore, we cannot make them into toxoids, right? We haven't been able to develop an endotoxin vaccine, which would be amazing, would save so many people's lives. And as we said, you guys, when the gram-negative bacteria are growing in us in a place they shouldn't, for example, if gram-negative bacteria are growing in our bloodstream, or if gram-negative bacteria are growing in our kidneys, or gram-negative bacteria are growing in our central nervous system or in the peritoneal cavity, as they die and break apart, they release the endotoxin, and that triggers cells of our immune system to release that cytokine storm, that overabundance of cytokines, right? And as we said, you guys, we need low amounts of cytokines locally, right, for a good immune response. But when we have massive amounts of cytokine being released, streaming throughout our bloodstream, we get these totally inappropriate responses that are that we describe with endotoxic shock, endotoxic sepsis. So remember, you guys, we get the vasodilation and leaky blood vessels that decreases blood pressure, decreases blood flow to our tissues and organs, right? That's the hypotensive shock. We get DIC, we trigger the clotting cascade, so now we're throwing blood clots everywhere. And the combination of hypotension, DIC, you guys, will have multiple organ system failure because our tissues and organs aren't getting sufficient blood, leads to shock and can rapidly progress to death. We want to remember, you guys, that endotoxins are pyrogens. They'll trigger a fever. And an irony, you guys, this is so wild. In DIC, I actually saw this happen in a little dog that was in surgery undergoing a leg amputation. A little dog was thrown in DIC. And then what was wild, you guys, is following the DIC, the little dog used up all of its clotting factors. So then it started to hemorrhage out. It was just horrible. There are these little, almost like almost look like little red flowers under the skin. And that was where the poor little dog was hemorrhaging out through the capillaries because it had used up all of its clotting factors in DIC. And the amazing thing is, you guys, that little dog survived. They did really high level therapy, and they pulled that little dog through. But it was horrible to see it happen, right? And so the same thing can happen to your patients following DIC. They could end up hemorrhaging on you, right? Because they've used up all their clotting factors. So doggone it, you guys, if we could just get a vaccine against these endotoxins, that would be awesome. And even folks, and let's see. Oh wait, we'll go to the next one, right? So you guys, don't panic about this, but it's just showing you this sepsis, septic shock, how complicated it is. So do remember, you guys, like we've talked about, how the gram-negative endotoxin can trigger this inappropriate release of chemical messengers from cells of the immune system, and then we get this inappropriate immune response. Really, it's an inappropriate inflammatory response that can actually kill us, right? And do remember, you guys, it's not just gram-negative bacteria. Gram-positive bacteria can cause sepsis. And remember, you guys, the pandemic coronavirus, they think one way it's killing some of its victims is that the pandemic coronavirus in some people it's triggering this sepsis cascade, right? So the poor people, one reason they're dying is not only from the coronavirus infection, but their immune system, right, is responding inappropriately. And it's the immune system response, this cytokine storm, that contributes to the death of those poor people. And one thing too, folks, with, some people pull through sepsis or septic shock, but we know this from an acquaintance of our family that survived septic shock, but there was permanent damage. There's like permanent damage to the kidneys, so higher risk for kidney disease. And we think there was damage to the brain, right? Because the person it truly has never behaved the same following sepsis. So, um, yeah, man, if we could prevent sepsis, wouldn't that be amazing? And then, folks, just here, this kind of a concept map on sepsis. So the mortality rate can be 10 to 50%. So the worst case scenario is you're going to lose half your patients, right? And for those of you that are going into medicine, I find these concept maps fascinating. So I think H&P is history and presentation, right? So it's telling you all the signs that your patient could present with. if they're experiencing sepsis, general, respiratory, urinary, abdominal, CNS. And then over here, you guys, they talk about sepsis. And sepsis, it happens in steps, you guys. So we start out kind of with kind of like, okay, here's a point where you best be able to save your patient, and then we just keep progressing. Things get worse and worse and worse. So here we see the again, you guys, we were saying that the endotoxin is a pyrogen. The heart rate's going to be elevated, respiratory rate. And you guys, I'm so bad. I don't know these values. So you guys yell at me. I'm presuming the respiratory rate is increased, right? If you aren't getting enough oxygen to your tissues, your body's trying to compensate by increasing heart rate. Maybe you're like panting almost. And early on, at least, you should get an increase in your white blood cell counts, right? Often with gram-negative bacteria, get a neutrophilia, and then it progresses all the way to septic shock. And I think once you get to septic shock, folks, it's hard to pull people back without at least permanent damage. Over here, you guys, differential diagnosis, right? Here's treatment. So you can see a lot of it is supportive. So you push the IV fluids like crazy, trying to get the blood pressure back up. Medications that might help increase blood pressure. Antibiotics, of course, you're trying to kill the bacteria that are causing it. But remember, you guys, when you kill the bacteria with your antibiotics, they're going to release even more endotoxins. So you have to be prepared for that. Like, you might actually contribute to the sepsis. Steroids have been used. See, this one I don't know about, you guys. The recombinant human activated protein C, polyclonal, passive immunity, guys, antibodies, drainage and debridement, trying to get rid of the pathogen. Prevention over here, vaccination, gamma globulin, maternal group B strep, therapy, hand washing, catheter care, pathophysiology, just kind of the events behind this, right? And here's some of the causative organisms. And so you guys, they have gram positives, gram negatives, anaerobes, fungi, for example, candida. And do remember, you guys, with the pandemic coronavirus, it looks like it too can cause sepsis. I think that's it, you guys. So the next part of medical micro is epidemiology, reservoirs and transmissions. That will be the our first lecture next week, week 14. That's a separate PowerPoint. And so I'm going to be working on that this week, and hopefully I'll have it posted Sunday night or Monday morning. And then Monday morning I'll plan to do the audio with it. Okay? All right, you guys. So hope you all are doing well. We're almost there, you guys. We can do this. Okay. Take care.